So, hello everyone, and thank you for coming to the session of talks. As Rachel just said, today we're going to hear about a game of cellular telephone and how miscommunication and that telephone call can cause diseases. Um, we have three wonderful speakers lined up for you today. First up is Krishna Mudambi, who works in the Department of Pharmacology at Yale Cancer Biology Institute, and he works on timing of cancer signaling. And fun fact, he was a member of the Boise City Council in high school. Next up, we have Christina Cho, who works at the Department of Immunobiology at Yale School of Medicine. She does research on how tumors resist immune surveillance in our body. Outside of the lab, she loves to do yoga as, in fact, a certified yoga instructor. And last but not least, we have Erica Hoyos Ramirez, who works at the Department of Neuroscience at Yale School of Medicine, and she works on molecular mechanisms of neuronal communication or communication in our brains. And a fun fact about her is that she recently learned how to roller skate. So these three speakers are going to talk to you today about communication within our bodies. It sounds exciting, right? I'll let my fellow coordinator, Renuka, explain that a little bit more.
Thanks, Anandini. So before we go into our speakers' individual talks, let's talk a little bit about communication. So we all communicate on a daily basis, right? To our friends, our family, our coworkers, and we do this in several ways. It could be in person, over the phone, which could be a telephone or a cell phone, and even through email. And usually we communicate to convey some information. So if we think of a traditional phone conversation where there are two people talking through a corded telephone, there's usually a caller who makes this call and a receiver who picks up the call. So the information that the caller is transmitting travels through the cord and reaches a receiver. And what's really important for this to work is that the correct information from a caller goes to the intended receiver. Now, when this happens on a much larger scale, where there are many callers and many receivers, this can actually get quite complicated. But modern telecommunication systems have largely streamlined this process and ensured that even in a situation like this, caller and receiver pairs do not get mixed up. So similar to how many individuals are talking to each other in this room, our body can also be thought of as a room, where multiple lines of communication are happening on a second's basis. This communication happens between cells, and cells are the fundamental units in our body that perform all the functions required to stay alive. So our body is made up of trillions of cells, which are in different shapes and sizes, and they perform different functions depending on where they are present. So cells in the brain are often shaped like long spindles. Skin cells are cuboidal shaped, and cells in the muscle can be of different shapes depending on what function they are performing. So today our speakers will be talking about cells in the brain, cancer cells, and how normal cells communicate with each other. So in order for cells to perform and coordinate all their functions, they need to be able to communicate with each other. And they do this in a similar caller and receiver fashion. So cells can be thought of as bags, which hold many molecules. And when they want to communicate, caller cells send out a specific type of molecule called a ligand to the receiver cell. The receiver cell has antennas, which are called receptors, and they receive these ligands. And so when receptors on the receiver cell receive the ligands, they recognize them and bind to them. And depending on what pair they have, they perform specific functions that result in specific processes downstream of these interactions.
So imagine a world in which you could not communicate effectively. Things would fall apart, right? So similar to that, communication within the cells of our body, as Renuka explained, is important for us to perform daily activities and to stay healthy. So the three speakers are going to talk to you about this communication within our bodies. First up, Krishna is going to talk to you more about this, what this communication is and how these cells communicate with each other and how it's all about timing of the signal. When things go wrong in our body and we get diseases such as cancer, then we can actually alter the timing or fine tune the signal to make things right again. And then Christina and Erica are going to focus on two instances in which communication can go wrong. Christina is going to talk more about communication in cancer cells and how these tumors hijack the communication in our bodies for their own benefit and how immunotherapy can help. And then, Erica is going to talk about Alzheimer's disease. which happens when communication in a very important part of our body, the brain, goes wrong. So without further delay, I'll hand over the screen to Krishna, and let's see how it's all about timing.
All right, let me get my screen set up here. All right, thanks everyone for joining us. As the moderators mentioned, my name is Krishna Madimbi, and I'll be talking to you today about how cells communicate with one another and how we believe that timing plays a very important role in this communication process. So, as our moderators so nicely told you, we have trillions of cells in our body, and they're constantly in communication with one another. So the joke I always like to make about this is if you think, you're bad about phones, or your grandchildren or your children are really bad about being on the phone all the time, your cells are much, much worse. They're constantly making these cellular phone calls over and over again, right? Basically like texting forever. And it's really important that they do this because this lets them communicate with one another and let them know what's happening around them, right? Cells can understand what's happening around them locally, like their neighboring cells. More distant cells that are further away, for instance, like neurons, communicate with different muscles and things like that. And also, it's very important to know what's happening outside of the body. So these different communications give our bodies, our cells, different signals and tells them how to behave, right? It tells them what they need to do in response to this outside environment, the signals that come from there. So some of these things that they can do in response is they can grow. So we see here our cell growing up because of the signals it's received. Certain signals can tell our cells that now it's time to divide. Other signals can tell our cells that it's time to break down and digest food, a process we call metabolism. And another really important signal that our cells need is they need to know when to die. So there's specific signals that come in and tell our cells that now it's time to die. And when there's a miscommunication between cells and these signals are misinterpreted, we see the rise of various diseases, right? Cancer is a really great example, particularly when we're talking about cells not dying appropriately, right? If a cell receives a signal that tells it to die, but instead it misunderstands it as a signal to grow, we can think of how that could contribute to something like tumor growth. So let's zoom in a little bit on our cells here and understand how this cellular phone call is made, right? So we're going to zoom in on these membranes. As our moderators mentioned, they're kind of like barriers that protect the cell from their outside environment. So it keeps everything that needs to be in the cell on the inside and keeps everything on the outside. So on our left, we have our caller cell surrounded by that protective membrane. It contains the signal or ligand that's going to be sent out to our receiver cell here on the right, which has those special antennae or receptors that can bind to specific ligands. And this initiates the receptors coming together and starting a signaling process, right? So now that outside signal is going to be relayed into the cell. And once that signal is relayed into the cell, it's going to interact with our DNA and basically turn on or off different parts of our DNA called genes, which code for different functions, right? So if we take our hypothetical DNA here, this section might code for cell growth. So that might be a gene that's turned on. Another section or another gene might code for cell division, and still another might code for cell death. So these are different genes that can be turned on and off, and regulated very specifically to get a specific cellular outcome in response to our outside environment, the signals that we receive from there, right? So let's zoom out and take a look at this whole process, right? So let's hypothetically say that our cell receives a signal that says, hey, you had a good run, it's time to die, walk to the light, right? So this signal is going to come in the form of a ligand. It's going to bind to a specific receptor that turns on the genes that tell the cell that it's now time to die. And you can think about how this has to be very finely regulated, but there can also be problems where this communication gets misinterpreted, right? As we mentioned earlier, which causes diseases. And this happens through various mutations. And you're probably familiar with the idea of mutations these days, given all the news that we've heard about COVID-19, right? We've had one specific strain of COVID-19 that started, and now over the last year, we've seen various different strains that have cropped up because there are mutations in that spike protein of the virus, right? The spike protein are those little protrusions that come out of the coronavirus. And these different mutations have changed the way that this virus behaves, right? It might make it more infective, make it more virulent, more difficult to stop with various vaccines and whatnot. And likewise, cancer mutations are similar. The receptors that bind to these different ligands can be mutated so that they behave differently. Those signals that originally come in for something like cell death might be misinterpreted for something like cell growth. So let's once again go back to our hypothetical caller and receiver cells and see how this works, right? If we have a signal that's sent by a receiver cell, or a caller cell, and it binds to these mutated receptors that no longer function properly, a signal like cell death might be misinterpreted as something like cell growth. So you see there's a change in the signaling outcome, right? For instance, the signal might be amplified in a different way to turn on the genes for cell growth or cell division. Now, the way we've studied this has really determined how we've tried to target these problems in therapy, right? And what I mean by that is, let's take an example of some blind scientists trying to study an elephant, right? Depending on what part of the elephant the scientists are studying, they may have a different interpretation of what that animal actually is. So for example, this guy here on the bottom right, he's studying the trunk of the elephant. And by looking at the trunk, he might think that the elephant is actually a snake-like creature, right? Likewise, the scientist on the top is looking at the ears of the elephant. He may think that it's more like a fan-like creature. The scientist on the left that's measuring the tail might think that the elephant's actually more like a rope-like creature. And further still, the scientist that's on the elephant might think actually it's more like a big wall, right? And they're not all necessarily wrong, but they're not talking about the whole creature as we think of it, right? They're talking about bits and pieces, which all make up the elephant. They all have special functions, but they all help the elephant do whatever elephants do. They don't actually make up the elephant, right? They're individual pieces, part of a whole. And likewise, we've tried to piecemeal study signaling as well. Many people spend their entire lives looking at just the ligands, or some people look at ligand receptor interactions, and other people look just at gene regulation, right? Whether things are turned on or off, whether these genes are turned on. And based on what they've studied, they've tried to create different therapeutics to target just those functions and turn them on or off in response to treating cancer. And we all know that this is not the best way to do it, but unfortunately, given the limitations of technology, we have to somewhat focus in on different regions. But now as technology improves, we're starting to back away from this and try and understand the process a little bit more holistically. So let me give you an example of how people have tried to treat lung cancer, right? Lung cancer has often been focused on this ligand receptor interaction. And what we've done over the years is we've tried to just basically block bad signaling. We know that miscommunication causes cancer, so our idea has been, hey, let's just stop the signaling, let's stop that communication, we'll stop cancer, right? And to do this, we've created different therapeutics that, for instance, stop things like ligand receptor interactions, right? We've tried to just stop that so that the signaling can stop. We've further tried to stop the receptors from coming together and initiating signaling. And we've also tried to target the internal part of the receptor that starts the signaling into the cell, turning on various genes, right? With the whole idea being that, hey, we need to just stop this bad signaling because it's giving us cancer, right? It's giving us a bad result. The cell is misbehaving, so let's just stop the whole thing. And while this might sound like a good idea, unfortunately, it hasn't worked as well as we'd like. And this is because there's lots of resistance that comes up to cancer treatments when we try and inhibit or block the signaling. And this happens because further mutations happen to the receptor that make it not only cancerous, but also resistant to cancer treatment. So let's take our hypothetical person with lung cancer. If we see a patient come in with lung cancer, we'll often try to diagnose it and then treat it with a first line of lung cancer treatment, right? Some inhibitor or drug that tries to block signaling. And this is usually pretty well received by our patients. We see an increase in survival rate for them. They live for another year or two. They respond quite well to the treatment. But after some time, many patients then become resistant to that cancer treatment, right? They no longer respond to the drugs that they're given, and we see a resurgence of that cancer. And when we, again, do a biopsy of these patients, we typically find that there's new mutations that have risen that are now resistant to the cancer therapy that we've given them. And to combat this, we try and then give them a next line of lung cancer treatment, something that's maybe mutant specific. And this also works for a little while. We see patients respond well for another year or two, but then again, new resistance mutation pops up and we basically repeat the cycle, right? You can imagine how a new resistant comes up, We try and treat that resistance with new drugs. Another resistance comes up. And this is pretty much untenable, right? We can't do this forever. We just don't have the drugs available to treat every new mutation that crops up. So one of the big questions in my field is, how do we combat this resistance? How do we prevent cells from resisting the drugs and therapy that we give our patients when it comes to something like lung cancer? So in my lab, we've started to really reevaluate the whole signaling process, right? As I mentioned, this idea of trying to stop or shut down signaling doesn't seem to always be helping. So we started to think about other things that we can target. Is there something besides these interactions that we can target? Is there something we can do besides just shutting them down? And recently, we realized that timing of signaling is very important. So what I mean by that is different ligands cause different receptors to signal in different durations, right? For instance, this purple ligand allows these receptors to come together for a long period of time. We have a long interaction, and that tells the cell, hey, it's time for us to become specialized or reach a finalized state where we no longer divide and grow. And this is going to be what our new function is, right? Now, the same receptor can have a different ligand, here shown in gold, that can cause a short interaction or a short signal to happen, which tells the cell to do something completely different. That short interaction or short signaling might tell the cell, hey, it's time for us to divide and grow. And you can imagine how this division and growing is pretty bad for cancer, right? So we started to think about how we can really understand the signaling so that we can start to create new treatments or therapeutics to target the signaling as opposed to blocking interactions. And to do this, we realized that we need to observe these receptors at the molecular level. We need to study them individually, one by one. And this is very difficult to do because cells themselves are too small to see by eye, and these receptors are even tinier. You can think of them as like 1/1000th the width of your hair. So they're super tiny. And the way that we visualize them or look at them is by sticking what you can think of as molecular light bulbs onto these receptors. So here I'm showing two receptors. One is tagged with a green light bulb. Another one has a purple light bulb affixed to it. And when we do this, we can watch as they interact and come together. And then given the properties of these light bulbs, there's a color change when they interact together, right? And we can see how long this color change happens, and we can track it over the cell and really get a fine idea of the timing that's happening between these interactions and really start to understand how signaling works. So what I'm going to show you here is actually that cartoon as a real experiment. So this is a real experiment that I do day to day, where I have different cancer cells that have these receptors, and I put on these molecular light bulbs that I mentioned. So one is shown here in green. I have one shown here in purple, and we're going to watch how these receptors move around on an individual cell, and we're going to watch them come together and interact. So here you can see in a live cell, these things are pretty dynamic. They move around. And when they're by themselves, they're their individual colors, green and purple respectively. But we can also see when they come together, we see a change of color into a white color. And we can now look at the timing of that color change. How long does it stay changed? How long does it take before they break apart, to really understand how these receptors behave when they're signaling? And as you can imagine, this is the forefront or the frontier of these kinds of studies, right? We're really starting to understand signaling in the sub-second level, right? We're really understanding them below 1 second. And we can also start to understand how these receptors behave when we change the signals that they receive, when we change the ligands they interact with, when we give them different types of cancer therapeutics. We can study these receptors and see how that timing changes as we change these different conditions. And this starts us thinking on the pathway of, well, what if we can fine tune the signaling? Now that we get to know the timing, is there a way we can alter the timing so that we have a behavior from the cells that we like? So if we go back to our example of a long and short interaction, right? Long interactions of receptors allows the cell to reach kind of this finalized state where they become very specialized and they no longer divide and grow. Short interactions, on the other hand, right? They can cause the cell to basically divide and grow. So we started to wonder if we can fine tune these short interactions with drugs that actually lengthen the interactions, right? So if we give them a new therapeutic that changes that short interaction into a long interaction, can we start to change the way the cell behaves? Instead of growing and dividing, can we then tell the cell, hey, actually, let's become specialized. Let's stop growing and dividing. Let's reach this kind of finalized state and basically no longer be cancerous, right? The idea here is to take problematic cells or bad cells and turn them into good cells where they're no longer behaving in a cancerous way. They're not just continually growing and dividing. So we really hope that this is going to be the way that we start to think about cancer therapy in the near future. And I'm hoping that these kinds of drugs and therapeutics will reach our patients in the next 10 to 30 years and really effectively battle cancer and hopefully lengthen the lives of our patients and also give them more favorable outcomes than we're seeing with the current therapeutics. So let's quickly summarize what we talked about today, because I think it's also going to be very important during Christina and Erica's talks as well. So I think it's important to keep in mind here that cells are in constant communication because they need to understand what's happening in their outside environment, and they need to realize how they have to react to that outside conditions, right? How do they divide? Do they grow? Do they die? And when we have miscommunication in this cellular telephone game, we see the rise of diseases, right? So I just mentioned cancer, and we're going to hear about a couple of other diseases as well. And another thing we've talked about is how the current treatments for cancer particularly haven't been completely effective, right? When we try and stop or shut down signaling, we often see that there's new mutations that arise that make the cells resistant to this kind of treatment. So in my lab, we're starting to try and think about how we can alter timing instead to make bad cells turn good, and thereby creating just better cancer therapeutics. So with that in mind, I'm going to hand it off now to Christina Cho. She's going to tell us a little bit more about cancer and how cells can disguise themselves and basically escape detection by the immune system. I'm going to hand it over to her now.
Hi, everybody. All right, thanks, Krishna. That was great. So today, well, I'm Christina, and today I'll be talking about communication inside the tumor microenvironment or inside solid tumors. And so cancer cells live in what we call a diverse tumor microenvironment. So when we think of a tumor, a lot of people might think that it's just a bunch of cancer cells, but it's actually not just cancer cells. A tumor is a large mass that contains cancer cells, different types of normal cells, and non-cellular components that help maintain the shape of the tumor. So if you look at this diagram right here, this is a picture of a tumor. So you'll have cancer cells, obviously, but you also have these normal cells. One type of normal cell is called the cancer-associated fibroblast. And another cell type that I'm sure you guys have heard of in many other contexts are immune cells. So immune cells, cancer-associated fibroblasts, and several other normal cells live together with cancer cells in this microenvironment. Now, the cells in the tumor microenvironment communicate with each other, much like we were talking about earlier, about cells constantly talking to each other to make sure that they're doing the correct functions downstream. In tumors, the cells communicate with each other so that the tumor either grows or stops growing. And so cancer cells communicate with each other. Cancer cells also communicate with immune cells, but cancer cells can also communicate with cancer-associated fibroblasts. So the first part of my talk, I'll be discussing the interaction or the communication between cancer cells and immune cells. So the immune system is the human body's defense against cancer. If we were to think about the human body as a nation, the immune system is like national security. And like our national security has teams and divisions, there are also teams and divisions in the immune system, and it's tightly organized, and there is a chain of command. So you start with the reconnaissance team, right? In national security, they go and look for foreign threats, things that could harm our nation. And they let the special forces know, hey, look for this threat, it must be terminated. And the special forces will go and look for that threat, terminate that threat. And then the resolution team will come in and say, hey, you've done your job, stand down, you're discharged, we're good to go. In the same way, the human body's immune system has several teams, and each team has their function. So the reconnaissance team is made up of what we call antigen-presenting cells, and the special forces include a special type of immune cell called the killer T cells, and the resolution team are what we call regulatory T cells, or Tregs, and they all communicate with each other so that they can make sure that your body stays healthy and void of foreign threats. So let's take this analogy and talk about the chain of command. So first you have a foreign threat, and this threat is recognized by Homeland Security, which then sends out your special forces to go and terminate that threat. In the same way, your cancer cell is detected by the antigen presenting cell, which then lets the killer T cell know what to look for and who to get rid of. So I want you to take a careful look here at the word antigen. You guys might have heard of this word, but if you haven't, I'm going to tell you what it is. So an antigen is a distinguishing marker that lets the immune system know that something is foreign. A really good analogy to this is an area code. So when someone calls from an international number, you know that it's an international number by its area code. We also know if a call is coming from a local region or out of state by its area code. The same way, foreign objects have these markers that let the immune system know that doesn't belong there. Foreign objects like viruses, bacteria, and cancer cells express antigens that are recognized by these antigen-presenting cells. And so here I highlighted viruses because I'm sure you've heard of the word spike protein several times in the news, and that's a type of antigen. And so cancer cells also present an antigen. So the cancer cell presents an antigen that's recognized by the antigen-presenting cell or the reconnaissance team. The antigen-presenting cell then presents that antigen to the killer T cell to let the killer T cell know exactly what to look for, what marker to look for to make sure that it kills the proper cancer cells. And then the cancer cell goes and looks for that, the killer T cell goes and looks for that cancer cell and terminates the threat. And that is how your immune system communicates with each other to destroy tumor cells. And the immune system is tightly regulated. So all of this communication happens under tight control. And this is to prevent chronic activation of the immune system. Chronic activation of the immune system can cause other diseases like COPD, asthma, all sorts of things that you don't want to be going on. So the Tregs have to come in and let the killer T cell know, hey, the threat has been cleared, stand down. And in this way, you avoid chronic activation. So the Tregs communicate to killer T cells using immune checkpoints. And these immune checkpoints are what truly regulate the immune system to prevent chronic activation. Immune checkpoints are molecules or ligands that we talked about earlier that signal to the killer T cells to stand down. A really good analogy for that are code words. So the Treg will give a code word that is recognized by the killer T cell as a signal to stop killing, to calm down. An example of this immune checkpoint is the PD-L1, PD-1 signaling pathway. So these work as immune checkpoints. So the Treg will say PD-L1, and in response, the killer T cell will say PD-1 code accepted. And that communication is what terminates the killer T cell activity and allows the immune system to shut off. So I'm just going to break this down a little bit more. PD-L1 is a ligand that is expressed on Treg cells. Earlier in our talk, we talked about ligands and receptors and how they interact to cause signaling in the cells. And that way the cells know how to behave. So the PD-L1 is a ligand that is expressed on Treg cells. PD-1 is the receptor on killer T cells. And when PD-L1 and PD-1 interact, killer T cells are deactivated and the immune system can calm down. So again, code PD-L1 is given by the Treg cells and the killer T cells in response will say PD-1, code accepted. Unfortunately, cancer cells have learned to hijack these immune checkpoints to hide from killer T cells. So under normal conditions, you will have a threat that's recognized by Homeland Security, and the termination team or our special forces will go and get rid of that threat. And once that threat has been terminated, that confirmation is given, and the termination team or the special forces can now be discharged. However, in the case of cancer, the cancer cells have hijacked PD-L1, that code word. And what ends up happening is the killer T cell is prematurely shut down and it can't do its job of terminating and killing the cancer cells. So the cancer cells continue to grow and divide and the tumor can keep getting larger. So what immunotherapy tries to do is it tries to fix that hijacking. Immunotherapy is a new type of cancer treatment that is a little bit all over the news right now because it's this hot new type of treatment that is FDA approved for certain types of cancer, including melanoma and lung cancer. So Keytruda and Opdivo are two therapies that are currently available for the treatment of certain tumors, and they are antibodies against PD-1. Antibodies are proteins that bind to the antigens, specific antigens, and shut down whatever activity comes after comes down after the receptor is bound to, the ligand is bound to the receptor. So if we go back to the ligand receptor interaction, the antibodies that are against PD-1 bind to PD-1 to prevent the cancer cell from interacting and shutting off killer T cell activity. And that way, these antibodies allow the killer T cell to continue to do its job and terminate the tumor. And these antibodies are actually have been developed over the course of the last 5 years is where the therapy has really improved. And what's really cool is that the lab that I'm in currently, my mentor is actually one of the key people who identified and discovered this checkpoint pathway and helped develop these therapies and initiated the initial clinical studies that really helped develop these therapies against cancer. So over the course of the 5 years, as this therapy started developing and getting better, we started noticing that although patients who received immunotherapy tended to do better, so their tumors did get smaller and they lived longer, only a minority of patients actually benefited from this therapy. So the patients that benefit from this therapy, we call responders. So responders are people whose tumors, once receiving immunotherapies, shrink, and it overall improves their health. However, the majority of patients are non-responders. So even though they receive the immunotherapy, their tumors do not shrink and their health is not improved overall. And so the current question in the field is why? Why are the majority of patients non-responding? And what makes the responders respond? What is unique about this population of people, and what can we learn about them to help us improve the efficacy of these new drugs? So for a clue, we go back to the tumor microenvironment. As I mentioned in the very beginning of my talk, I said that the tumor microenvironment is made up of many different kinds of cells, including normal cells like cancer-associated fibroblasts and immune cells. Well, we just talked about what the immune cells do. I'm going to talk briefly about what cancer-associated fibroblasts do, because this is where my research really comes in. So cancer-associated fibroblasts are a normal type of cell that lives inside the tumor, and it has many different functions. It produces different kinds of proteins that help cancer cells continue to grow and divide. It also has different proteins that affect how immune cells function. Cancer-associated fibroblasts also produce the non-cellular components that help maintain the shape of the tumor. So these fibroblasts are very special, and they do many different things to help the tumor grow. One of the things my research is starting to uncover is that cancer-associated fibroblasts directly and indirectly communicate with immune cells, effectively shutting down their function. So they're no longer able to do that chain of command to either detect the antigen by the antigen presenting cells, or the killer T cells are not active or not functioning properly. And that signal, that communication here is what my research is trying to uncover to figure out exactly what message the cancer-associated fibroblast is giving to the immune cells so that they don't work the way they're supposed to. So let's quickly go over the main points of my talk. So your immune system has the ability to detect and kill cancer cells. So the cancer cell presents an antigen that is recognized by your reconnaissance or surveillance team, your antigen presenting cell. The antigen presenting cell then presents that antigen to the killer T cell, your special forces, which then go and get rid of that cancer cell. This whole system is then regulated by the Tregs, the calm down team. I can't think of the name all of a sudden. The resolution team. The resolution team comes in and lets the killer T cell know that the threat has been terminated and to stand down. And it does that through immune checkpoints. Unfortunately, cancer cells have figured out a way to hijack those immune checkpoints and hide from the immune system. So the cancer cell expresses PD-L1 to effectively shut off killer T cells before the killer T cell has completed its job. However, immunotherapy comes in and blocks this hijacking to reactivate the killer T cells to let them finish removing all the cancer cells and shrinking your tumor. So to summarize, what we've talked about today is that when communication between immune cells goes right, when that chain of command is working properly, the tumor shrinks and the disease is stopped. But if communication between immune cells goes wrong or gets hijacked, the tumor can continue to grow and cancer cells can continue to divide, and you end up having worse disease. So the question now is, what happens when communication goes wrong in other parts of the body? And that's where Erica will come in, and she'll talk about what happens when communication of cells goes wrong in the brain.
Thank you, Christina. That was really great. Let me share my talk. Right, can you guys all see that? All right, so thank you all for being here. Great, thank you. Thank you for being here today and for listening to our science talks. As Christina mentioned, I'm Erica Ramirez. I'm a neuroscientist at Yale, and I will tell you all about communication inside our brains and what goes wrong in Alzheimer's disease. So we heard from Krishna how cells communicate constantly to pass on information about the environment, and how, if we understand how the timing of this communication occurs, hopefully we can create new drugs to target cancer. And Christina told us how the immune system fights disease with very tightly regulated messaging systems, and how tumors can hijack those systems to hide from our immune system. We are now going to look inside our brains. Our brains are composed of many different types of cells. But the very special kind of cell that lives in our brain is the neuron. Neurons have specialized structures that allow them to act as electrical cables. And by using electricity, neurons can transfer information at very fast time scales and allow us to perceive the world very fast and behave and respond to the environment in a very fast way. So let's look inside our brains and let's see how neurons communicate with one another. We are going to start by looking inside an intact brain. If we could clear all the tissue and all the different types of cells that are in the brain and only look at one type of neuron, we will look at something like this. We can clear the tissue of an intact brain using mouse. Mice are very important for neuroscientists because we can use them to understand the complexity of the brain in a real life animal. So we have engineered these mice to make the neurons shine, and each shiny dot that we see here is the main body of the neuron. We can see how neurons project these cables, very long cables, that act as electrical connections that allow neurons to communicate with one another across different brain regions. So let's look deeply into the neurons. If we zoom in and look at a cluster of neurons at very high resolution, we actually see that neurons resemble a lot like little phones, functionally. Neurons receive all these messages in these very tree-looking antenna projections. And that's where all the phone calls and all the signals from all the neighboring neurons are going to communicate with ourselves. Now, all these signals will have to travel through these very long cables and reach the main body of the neuron. The neuron, that main body acts like a computer, is going to integrate all the signals and is going to tell the cell what's happening in the environment. And if the cell decides that it has to make a phone call to the other cells to relay that information, it will do that by sending a very fast electrical current that will travel through the cables and reach our other cells. So if we look at this process in detail, and now we're going to zoom in to the molecular level. So what are the molecules doing? Our neurons are very smart because they don't receive all the phone calls at the same location. They have these little tiny protrusions where they isolate individual phone calls. So each phone call is going to happen at these protrusions, and that's what we call the synapses. And if we zoom into that, we will see that when our caller cell releases an electric shock, that electric shock will release the signal of our ligands or the molecules. Those molecules are going to bind to receptors on the receiver cell that are located on that little protrusion. So when these receptors are activated by these molecules, some of those receptors are going to generate charge, electrical current that is going to travel through the neuron and is going to tell the neuron what to do at very fast speeds. But there are other types of receptors there as well. And there's these kinds of receptors that when they bind to these molecules from our caller cells, they are not going to generate electricity, but they are going to generate chemical reactions, changes in the biochemistry of the neuron that are going to affect the long-term behavior of the neuron. So let's see how these two elements of how the neurons communicate play a role when we are learning new memories or learning new information. And we're going to take an example of a song. Imagine that you are hearing a song and that all those sound waves are going to be transformed into electrical signals by our brain cells. And that electrical signal is going to target our neuron cells that are going to respond to the music. And they are going to generate some electrical currents and chemical changes that will allow us to be aware of the song. Now, the resulting changes of these chemical and electrical interactions in many different parts of the brain will determine whether you like the song or not. Now, assume that you do like the song, and now you hear the song again and again. What is happening now is that our caller cell is making that phone call many times and repeatedly. And what happens with that is that now our electrical signals and chemical responses become very large. And the outcome of those very large responses is that our neuron is going to start putting more and more receptors on those membranes to listen to that song. It understands that it's important information that it likes, and so it's going to put more receptors there so that the next time you hear the song, now our cell is going to respond with a very large current and large chemical signal. So that's, in a nutshell, what we understand at the cellular basis for the generation of memories. That's how neurons encode new memories. We are going to look at what happens in Alzheimer's disease and why patients that have Alzheimer's disease are no longer capable of retaining new information. What is happening at this molecular level? So Alzheimer's disease, as you may know, is the most common form of dementia and is also the 6th leading cause of death in the US. It is characterized by a progressive loss of cognition and memory, and in the latest stages of the disease, patients with Alzheimer's disease start to lose awareness of their surroundings and inability to be aware of recent experiences. Even though we as neuroscientists still don't know exactly what causes Alzheimer's disease, we know that it's characterized by two main features. The first one is what we call neurodegeneration, and that just means that the neurons start to die. And if we compare the brain of a patient with Alzheimer's disease against the brain of a healthy individual, we see that the brain from the Alzheimer's disease patient has shrunk and has lost a lot of neurons. Another feature of Alzheimer's disease is what we call the beta amyloid plaques. So beta amyloid is a molecule that lives in our brain and that plays a very important role in how the brain works. But somehow patients that have Alzheimer's disease start generating a lot of this molecule. And when we generate a lot of beta amyloid, that molecule starts to accumulate and generate these plaques around the neurons that becomes toxic and makes the neurons die. So let's see what is beta amyloid and how it has to do anything with brain communication, communication between neurons. it turns out that in the location where neurons communicate, the synapses, right, we have our receptors that are important to tell us what is happening and are important to tell us how to respond. And there are molecules. One of them is this long looking molecule that we call the amyloid precursor protein. This molecule can be cut when the cell is activated. It can activate systems that can cut this amyloid precursor protein with different type of chemical scissors. If it is cut with the alpha scissors, it generates this fragment, yellow and brown, which is okay. But if it is cut with the beta scissors, it generates this fragment, which we know is the beta amyloid. So this brown and blue fragment is that beta amyloid fragment. Now, this beta amyloid ordinarily in the brain will go to the color cell and will tell us, maybe don't call me so much. It's going to regulate how much the phone call happens, the regulation of the phone call. But if a lot of this beta, the scissors to this beta molecule happens, then we have a lot of beta amyloid forming, and that generates toxicity and the plaques. And then now we have cut the communication between neurons. And when neurons can no longer communicate, then they start to die. So is there a way in which this beta amyloid can be formed or not formed with the communication between neurons? The answer is yes. When our color cell sends a signal, the receptors that are there ready to receive that signal can trigger those chemical reactions that will cut the amyloid precursor protein with the alpha scissors. But some other receptors can also induce the cut through the beta scissors. So if there is a way in which we can tweak neuronal communication so that we can remove the beta cut and only process the amyloid precursor protein with the alpha scissors, that could improve the outcome of Alzheimer's disease patients. So that is indeed what current therapies are doing for Alzheimer's disease. The most common therapy that there is for Alzheimer's disease patients right now is what we call the acetylcholinesterase inhibitors. So you may have heard of it or may hear about it in the future. But all what this does is a molecule that we can give to these patients that are going to enhance the signaling of the neurons through these receptors, through the receptors that induce the cut with the alpha scissors. Now, this therapy has shown to improve cognition and also diminish the amount of beta amyloid that is present in these patients. But long-term use of these inhibitors triggers compensation of the neurons, where neurons do no longer make the molecule that's activating these receptors. So an alternative therapy that is being developed now is to create external molecules that are very similar to those ligands that are secreted by our color cell, and that they can bind very specifically to these receptors that enhance the cut through alpha scissors. And if we enhance that signaling to alpha, then we decrease the signaling to beta, and therefore decrease the amount of amyloid toxicity. The alternative therapy for Alzheimer's disease is to use immunotherapy. As Christina told us, the immune system is a very powerful machinery that can fight disease with a very specific messaging system. So if we can give our patients, Alzheimer's disease patients, a tiny little fragment of that beta amyloid protein, so that is long enough that it can train our immune system to recognize the bad molecule and plaques and remove those plaques, remove the toxicity by those plaques. That way we can improve the outcome of the patients by reducing the beta amyloid toxicity. So this beta amyloid immunotherapy is currently in clinical trials, and we are hoping that we can use that as an alternative therapy for Alzheimer's disease. So with that, let's see what we have seen today in our game of cellular telephone. Christina told us how if we can understand the timing of this communication between cells, we can fine-tune the signals so that we can tune bad communication into good communication to prevent cancer. Christina told us how immunotherapy can block the hijacking of the immune system by the tumor cells and that we can use this immunotherapy to enhance the response for the immune system to fight certain types of cancer. And I have told you how we can tweak how neurons communicate to find new cures for Alzheimer's disease. And with that, I would like to thank you all for your attention. A special thanks to Sunandini, Renuka, and Janki for putting this talk together. And we can open for discussions or any questions that you may.
Thanks, Erica. So I think we have the first question that is directed to Krishna, I believe. So Iris asks, are you using human cancer cells, and are they generic cells or are they specific lung cancer cells?
Yeah, so this is a really great question. Right now, most of the work that we're doing is on cancer cells. We do have plans to expand to different types of cancer. It just so happens that cancer cells are the easiest to work with. So that's kind of where we're starting these studies. And since they're very initial, it seems like a good place to start. But yeah, the plan is definitely to expand to other cell lines, other cell types, modeling different cancers and things like that.
Thank you, Krishna. So we have another question from Iris, and this is directed towards Christina, I believe. So do the cancer cells make the fibroblast, or do they hijack existing fibroblasts or cells and transform them?
Are you a scientist? That's a good question. And
I'm a kindergarten teacher.
You're a science teacher?
Kindergarten.
Kindergarten teacher. I think you're a scientist. So actually, the cancer cells do, what they do is they produce certain things that can change fibroblasts that are in the area of the cancer cells into what are called cancer-associated fibroblasts. So what we say in the lab is that fibroblasts are pathologically activated into cancer-associated fibroblasts. And so in a way, cancer cells make them, but it's not exactly clear how they are making them. There are a couple of clues. I published a paper last summer about one way it does it. It's through the production of certain cytokines. However, there are several different papers coming out that look at how cancer cells help make the cancer-associated fibroblasts. And also the second part of the question where you're asking about transformation, that's exactly what we call it also. There's a transformation of certain normal cells into these cancer-associated fibroblasts. And that process is really characteristic of very aggressive bad tumors. When you see not just a lot of cancer cells, but a lot of cancer-associated fibroblasts. So for example, in pancreatic tumors, like 94% of the actual tumor mass is not cancer cells, but the cancer-associated fibroblasts and the non-cellular components that are made by those fibroblasts. So that's a really good question.
So we have another question from Iris. Thank you so much. You have questions for everybody. So this is for Erica. Does the beta amyloid have a purpose?
Yeah, that's a great question. So yes, the beta amyloid is important in regulating how much that colored cell can activate the receiving cell. So it can regulate how much the signals are released. And that is important for exactly avoiding toxicity. Too much of that signal is formed that could be bad. So under normal circumstances, beta amyloid is important. But the problem in Alzheimer's disease is that it starts to form in large quantities. And now the body doesn't know what to do. And what it starts happening is that instead of helping regulating that signal, it just completely shuts down the signal by two ways. One is that it prevents the color cell from actually releasing the molecule that is needed to make it a phone call. And by accumulating genetic toxicity, making outside of the neuron, the environment outside of the neuron, making it toxic. So it's no longer able to generate the chemical signals that it needs to communicate with others.
So we have another question for Christina from Elaine. Is there an immunotherapy application for autoimmune diseases?
So there are people in my lab that do study autoimmune diseases, and they basically use those. So the immune checkpoints that I talked about earlier, the immune checkpoints are really important in regulating the communication. So you do want the immune system to be active and to go and get rid of foreign antigens, things that are bad for you. But what you don't want it to do is be so hyperactive, it starts attacking self antigen, things that your body's supposed to have, things that that belong inside that are totally normal. But in the case of autoimmune diseases, your immune system is attacking self. And so what people in my lab are studying are how to utilize those immune checkpoints. So activate those immune checkpoints instead of shutting them down. That's like the opposite kind of immunotherapy, so that it can control autoimmune disease. So yeah, immunotherapy is this wide range of different kinds of treatments. It's not just blocking antibodies, it's also activating agents that help signal in the right way. So if there's hyperactivity, you shut it down. If there's not enough activity, you activate it.
Thanks. So we have another question from Yankee to everybody, I guess. So any of you speakers can comment on it. The question is if you could comment on how the mutations of the spike protein found on the new strain of COVID could potentially alter cellular communication. Anyone?
Yeah, I could chip in here. So I think one of the things that we could see here, right, is if there's mutations on the spike protein that allow it to bind better to its receptor, the human receptor ACE2, it's more likely to trigger the basically taking the virus into the cell much more efficiently, right? So you might need less virus to actually have it go into the cell, more likely to do it. And I think something that's a little bit more along the lines of what Christina was talking about also is I think these mutations could potentially have it avoid being detected by your immune system, right? The shape of it might look differently than the antibodies that are specific, like let's say through a vaccine, right? The vaccine, the antibodies from the vaccine are for the normal looking spike protein, but these mutations might alter the shape in such a way that they're no longer recognizable by the immune system, so it'll act as if it had never seen these things before.
So we have one more question from Iris, and correct me if I'm wrong, was this directed to Erica when she was talking about beta amyloid proteins? The question is, so would immunotherapy prevent it from regulating the signals?
Yeah, it's a good question. Yeah, you may think so. You may think so that if you are now degrading all the beta amyloid, you can have the side effects of losing its actual function. Yes, you may think so. The idea, I think, is that you will give it to patients which are overproducing it, sort of to compensate for the overproduction, not to patients who are just making enough to use it as a cellular signaling mechanism.
Any other questions from anyone before I hand it back to Rachel?
Okay, well, thank you so much to all of the presenters for sharing your research with us. And thanks to the moderators, of course, and thanks to Iris for all the great questions. The program, as I said, was recorded and will be available on the Brookfield Library YouTube page. So you can watch again or share it with your friends. So thank you again to everyone for participating.
Thank you.
Good night.